Good morning, everybody. Okay, let me walk you through Viticast. Let me talk about some of the results that we have obtained so far from this project. So as for the source of this project, let me start by talking about what we do at Monet. This company was incorporated in 2014 in order to provide wineries and vine growers a solution for viticulture. So we have created a tool for decision-making purposes in order to monitor grapevines through a device that is connected to the internet. Our main line of research has to do with prediction models in order to identify potential fungal diseases in grapevines in order to reduce any uncertainty as to when the best time for treatment is. For developing, or in order to develop these projects, we used current literature, and we began testing these alternatives in different regions in order to improve the accuracy of these models. We included some new variables, such as, for example, different grapevine varieties, taking into account the characteristics of each plot of line. As a result, we came up with a new line of research, for example, for meteorological prediction purposes. In this regard, we have been working both in phenology and meteorology. We can identify climatic changes according to the different grapevine varieties. As for meteorological prediction, it's important to have local information at a plot of land level. Right now, our viticulture models take into account not only present and future weather factors, but also other climate variables. So what's the underlying idea of Viticast? We want to identify new variables in order to improve our prediction models, or we want to identify the severity of diseases taking into account phytopathogens in the environment. So we analyze spores that are present in the environment in order to create a relationship between spores and the severity of these spores. So here we more or less tell you what the project is about. So we do some data collection based on weather conditions. This is still in experimental phase. Then we also do some testing at a local level. We do some in-field observations. This is done by the phenology technicians and others that watch diseases, and then we have those in charge of collecting oospores that are subsequently tested and examined through microscopes at the lab. All that information is there afterwards entered into some AI models in order to determine the risk of a certain disease happening, and in order to determine the phytosanitary treatment that is most appropriate. What's the ultimate goal here? Well, we try to address a problem that is a true challenge for the viticulture sector. We are trying to fight against some endemic grapevine fungal diseases, such as, for example, powdery mildew, downy mildew, and botrytis that can cause serious problems to production. 3 years ago, about 80% of losses were reported, mainly in some regions where these diseases had not happened before. So in view of this problem, and since we are afraid of losing some crops, we sometimes see that some over-treatments are applied without taking into account meteorological conditions. So the use or the excessive use of phytosanitary products may build resistance against certain diseases, in addition to being an environmental pollution source while increasing production costs. So the Viticast pursues to provide top-notch solutions to fight against grapevine fungal diseases that are most common. And the main objective is to optimize the use of phytosanitary treatments in order to improve wine quality while protecting the environment and optimizing production costs. So in brief, we want to apply only a treatment if there is true and objective risk of a certain disease happening. In order to identify an infection risk, we use prediction models that we have developed, and we therefore add the phytopathogen related information based on what we have found in the environment. What about Viticast participants or stakeholders? Well, Monet Viticulture is the project leader. We provide know-how as well as AI models and disease prediction models and phenology studies. Then we have Feuga that provides its expertise in running projects as well as dissemination. Then Mata Romera, this is a winery that provides agricultural knowledge, and they also provide some plots of land for our experiments. Then the Pontevedra government also provides some support. We also have the University of Vigo support that provides its expertise and know-how on ospores. And we also have another subcontractor member. Speaking about a group of the University of Santiago de Compostela. Then we also have the Galician Viticultural Association and the Winemakers Association of Galicia. So the project is now being in progress. It has been in progress since November 2019. It's being run on 8 different experimental plots of land that are distributed in 4 appellation of origin regions, as you can see here: Rías Baixas, Ribeiro, and Valdeorras. Here we have set up an ospore sensor in order to gather ospores for the purpose of the project. We have also identified an area that is called Viticast. These are some facilities where the rest of the experiments will be run. So here we apply the typical strategies in order to draw some conclusions. In each of these 9 areas, we have selected a number of grape varieties, and in order to compare results between Viticast and the conventional area, we have created this configuration. Besides, in each area, we have created some soil sampling points in order to gather soil samples and in order to analyze chemical waste, both in the Viticast area and in the conventional area. Now, let me share with you some of the results that we reported in 2020. In 2021, of course, we do not yet have full results. As for downy mildew, first off, we calculated weather conditions in order to determine when the lifetime of the different of the fungus arises. So we apply a protocol. This is a physical protocol in order to determine the ripening of this fungus in the field. So we start calculating conditions in order to obtain some primary results. For example, last year we can see that the main disease or the primary disease occurred on March the 5th. Thereafter, we began calculating secondary incubation related to these diseases. For example, one was reported on March the 18th, and on the 20th, others happened. So the model was anticipated as a result of that. Once the primary incubation is over, we start analyzing sporulation, and then we also analyze secondary diseases and the secondary incubations, and this is a cycle that we repeat over and over again. As for the powdery mildew, we have developed 2 models, one that takes into account weather conditions and another one that takes into account both phenological and weather conditions. So as you can see here, there is one model that takes into account weather conditions only and provides a risk level that does not take into account the protection of all bunches, but the overall plant. So here the purpose is not to protect only the leaves and the grape variety, but actually the whole grape bunches. So we want to make sure that we cover the whole sensitivity phase. So here you can see some results So the straight line shows the results based on weather conditions, and the dotted lines shows the results by applying the phenology approach. So both models are quite similar if we compare the results both in May and June, but here on the 29th of June there was a difference of about 80% between both models applied. Well, in closing, let me touch upon some of the OSPOR model results. So we have to gather spores in the field. We have to analyze them through microscopes at the lab. As a result of this, we obtain the graphs that you see on the right-hand side. This chart shows us one of the neural networks that we have applied to developing the models. The inputs are weather conditions, and the output are the indicators of the risk of disease. So here you can see all the observations carried out by the technicians working in the field. So the overall results reported last year were classified into two groups. On the one hand, one group to draft the model and another one to run the test. So based on the test results last year, we obtained quite high successful percentages between 95 and 97%. So the system detects risk when it does not exist, and then we have dummy negative when no risk actually exists, when actually no risk is identified, but actually it exists. So in this case, we try to avoid the second scenario as far as possible. What about the impact and benefits that we expect from this project? Well, we expect to have some environmental and social sustainability, some significant reduction in the use of phytosanitary products in order to enhance the competitiveness of viticulture and therefore improve wine quality and less production costs. If you want further information about the project, you can visit www.viticast.es. Here you will find technical information as well as information about all the different activities we are going to host. I will be at your disposal to answer any questions that you may have, and here you have my contact information. Thank you very much.